12:15. My name is Jesper Wengel. I have the pleasure today of introducing our dear speaker, Himanshu Khandelia, from Department of Physics, Chemistry and Pharmacy. Is at Memphis Center for Biomembrane Physics. He's associate professor and has been here in Odense for a number of years now. Came first as a postdoc after his PhD, which he did at University of Minnesota. Then he became assistant professor, first postdoc, and then assistant professor, and now associate professor. He's a very productive researcher and one of the pillars of Memphis. He's obtained several grants, including a 10 million kroner Young Group Leader grant from the Lundbeck Foundation in 2011. Himanshu works on computational methods. He investigates membranes, proteins in membranes, and other fundamental molecular processes related to biology. So Himanshu, we look forward to your lecture, and the title is also very exciting, Biology on the Computer, even the supercomputer. So we look forward to your lecture. Thank you. Just so you know, let's take another sip from the court. Oh, I will survive. Thank you. I hope you survive as well.
I think I will. Okay. My voice is a bit hoarse. Can you hear me? Yeah. Okay. So thank you, Jesper, for the introduction, and thank you, DS, for inviting me here. In the next 35 minutes, I'll tell you about what you can learn about biology on the supercomputer. To start off, I'll show you a small movie. It shows a simulation box in black here containing some amphifiles, or lipids as we call them. And what you're about to see is a self-assembly simulation of some nanoparticles or aggregates that form from these lipids. This is what is typically called a self-assembly simulation, because we are not driving the self-assembly of these particles by any external forces except those that are inherently present in these systems. What you end up is with a small set of particles which are a few nanometers in size. I will talk about the physics of the movies in a little bit. We do simulations because simulations are now officially recognized by SDU as as the third way of doing science. So in the old days there was only theory you could do on pen and paper and experiments that you could do in the lab. But today, because of the huge computational power that we have, which is only going to get better, we can do a lot of useful simulations on the computer, so experiments on the computer. Here is a formal definition of a simulation from a colleague of mine who is not at SDU anymore. A simulation is a numerical experiment. So it's an experiment performed on a computer. If you can perform this experiment on pen and paper, then it's not a simulation, it's theory. Right? Only if you cannot do it on pen and paper and you're doing it on a computer, it's a simulation. Of the behavior of a model of a physical system. So we are not simulating a physical system, we are simulating a model of the physical system in which matter, and I'll come back to this later, is replaced by physical constructs. So matter is replaced by equations that interact in ways that mimic the real system and generates states equivalent to that in the real system. A typical example is a flight simulator or a video game. In a flight simulator or in a video game like World of Warcraft, you're sitting on a computer and you're trying to simulate reality. The difference is that a video game is considered to be very good if you simulate reality as closely as possible. This is what you want in World of Warcraft experience, right? You want to feel like you're really out there fighting with these beasts. At least some of us. But a simulation need not be like that. So in a simulation, the idea is not to replicate reality, but extract features that we are interested in from this reality using as simple a model as needed. Right? So a model need not be very complex. There are several reasons people do simulations. For me, simulations offer two things. One, visualization, which is underrated in physics and overrated in biology. And it offers access to properties not accessible to experiments, sometimes not accessible to experiments, which usually measure averaged out properties. I'll also come back to this. Often experiments are very complicated and difficult to interpret. There are too many variables. There is much greater control over a computer experiment. Even if you make a small mistake, if you run a large experiment in the lab and you work for weeks and you made a small mistake, you have to repeat the whole thing again. You can make the same mistake in the simulation, but at least you won't have to repeat the whole thing manually. But that's not what I meant by greater control over a computer experiment. In a computer experiment, you know exactly what you're doing. Your experiment will not be ruined because the test tube was dirty and not cleaned properly. For many tasks, simulations are cheaper and faster. Otherwise, there would not be a point of doing simulations unless we are learning something new. Okay, the talk is about biology on supercomputers. So let's talk a little bit about biology from the physical point of view. Long ago in 1997, I read a biochemistry textbook by Leninger. He's the father of biochemistry textbooks. And the first page of that line said, living things are made of lifeless molecules. I think that's a very profound statement. So the question the way we try to think about this is, how does biology, how do we as people arise out of these fundamental physical laws? This is one of the holy grails in science, I imagine. So right now, all of us, we are sitting here, you're listening to the talk, trying not to fall asleep. There are only two things going on in your body right now. There is either bond formation or destruction, or there are non-covalent forces. This is all we are composed of, unless you go into metaphysical or spiritual. So we'll leave that out, being at a university. But other than this, there is nothing going on. So you ate some of the food there that is being digested in your stomach. That's destruction of complicated starch into simple sugars. And we'll come to these non-covalent forces. Right now, your body is creating protein. It's destroying protein. Some cells are dying. Some cells are being made. All this is covalent interactions. But there are also non-covalent forces. And life, we claim, arises only out of these two types of forces. We are all familiar with the covalent bond. This is a covalent bond between two hydrogen atoms. Picture from Wikipedia. This is a covalent bond between two amino acids. For those who are not aware, amino acids are the building blocks of proteins in cells. So there are 20 different types of amino acids. All of them look like this, and this R group can be different chemically. So these 20 different types of amino acid form all the proteins in the cells, and these proteins are formally called the workhorses of the cell. They carry out many structural functions and catalytic reactions, and so on. So this reaction shows the peptide bond formation between this amino acid and this amino acid. So this is the new covalent bond that is right now forming in many of our bodies, in all of our bodies, I'm sure. But the shape of this protein comes about from non-covalent interactions. So for example, this famous alpha helix helical structure arises because of hydrogen bonding between certain atoms in the protein. And this ultimately leads to a certain shape of the protein, a certain conformation that is required for its function or structural function. In this case, we have an ion channel sitting in a membrane. The same thing is true for DNA. There are covalent bonds which form the structure of DNA, but non-covalent interactions between the two strands of DNA form its beautiful double helical shape. So what are these non-covalent interactions? And what is their magnitude? The important thing that I want you to remember from this slide is that non-covalent interactions are very weak in comparison to covalent interactions. So thermal energy is about 0.8 kilocalories per mole. This is the energy of fluctuations. The energy of a hydrogen bond is a little bit higher depending on which hydrogen bond it is. There is van der Waals forces. These are dispersion forces between atoms arising from dipole-dipole interactions. These are also very weak. And there are electrostatic interactions. So these are essentially, and hydrogen bond is essentially a type of electrostatic interactions. So these are all the non-covalent interactions that you have, other than entropy, which we should never forget. And all these interactions are about 100 times smaller than covalent bonds, and more importantly, their magnitude is the same as thermal energy. For comparison, if you have a pool ball hitting another pool ball, this force will be so many orders of magnitude larger than these forces. So what we are dealing with here are very, very soft forces in biology. And this is why simulations are important in biology. Because non-covalent interactions are of the order of thermal energy, they become very difficult to probe with experiments. Because your experiments fluctuate by thermal energy. And the time and length scales of many biological phenomena are very small. So these days we are interested in nanoscale or microscale phenomena and things occurring at a very, very short time scale. These again are difficult to prove using experiments, but fairly straightforward to do using the techniques that I will talk about in a moment. So a few slides about the method that we use. This is again a Wikipedia GIF. It shows gas particles in a box colliding elastically. So there are two types of gas particles, blue and red. You can think of these as nitrogen and oxygen molecules in this room. This is an MD simulation. Now imagine that you are able to measure the velocity of each of these particles in time or at different snapshots. So this particle is here now, in the next snapshot it is here, and for each snapshot you can measure the velocity of this particle. Then over a long time you can calculate the average, the mean kinetic energy of particles in this box, and that can be related to the temperature of the box. So the temperature of the box, which is a macroscopic thermodynamic observable which you can measure using a thermometer, can be described from the microscopic properties of atoms in the box. So the claim is that the thermodynamic properties of this box can be completely described by simply the positions and velocities of all the particles in the box. This is an important thing that you should remember whenever you see some movies that I'm going to show you later. Okay. So these, in statistical mechanics, these snapshots are called microstates. And a macrostate is what arises from these snapshots. A temperature arises from these snapshots. Right? So the temperature of this room now depends on the gas particles, the velocities of the oxygen and nitrogen molecules in this room. So if you could measure all this, in principle, you can measure the temperature of this room without using a thermometer. All right. So the macroscopic properties of the system can be completely defined by its microscopic description, which is essentially positions and velocities of all particles. And using laws of statistical mechanics, which is essentially mechanics, probability theory applied to mechanics, one can relate the microscopic properties of individual atoms to bulk thermodynamic properties of materials. So using such a simulation, if I do it carefully and if I do it long enough, I can in principle calculate these properties such as a viscosity or a dielectric coefficient or a diffusion constant or a binding free energy and compare it quantitatively to what you would measure in the laboratory. Okay, so to run the simulation now, all we need is a method to generate these microscopic states. So a method to generate positions and velocities. And the method that we use, there are several methods to do this. The method that we use in our lab is molecular dynamics, which is essentially solving Newton's second law numerically on a supercomputer or on a computer. So in molecular dynamics, you solve Newton's second law. So the force on particle I is the mass on particle I multiplied by the second derivative of its positions. That's the acceleration of the particle. And this is the same as the gradient of the total potential energy of the system with respect to the position of this particle. Now this energy function is usually very complicated, and therefore you cannot solve these equations analytically on pen and paper like you have done for simple things in high school, like two cars approaching each other at this speed and this acceleration and so on. Since this energy function is very complicated, we use numerical integration. So we choose a small time step, which I call delta t later, during which I assume the forces are constant. And we propagate the equations of motion in like this. So if this were the real trajectory of the system, you would approximate that using a numerical integration like this. The output from the simulations are positions and velocities of all particles in time. Are there any questions so far? A little bit about this energy function E. This is a typical classical force field. When I say classical, we are saying we're ignoring all quantum effects here. Not ignoring them, or we're not ignoring them, but we are somehow incorporating them in our simulations. The classical force field typically looks like this. So for example, the potential energy of vibration of two atoms around a bond can be approximately or quite accurately described by a simple harmonic spring. Yes. So here I want to come back to the original definition of the simulation. I said that the simulation in the simulation, the real system is replaced by equations. So here a bond has been replaced by a harmonic spring. And this harmonic spring will behave very similar to a real bond. So this is the energy of two particles vibrating around a single bond. You have a similar term around for angle vibrations, a similar periodic term for dihedral rotations for four particles which are bonded to each other. And then there are interactions between particles which are not bonded to each other, and these are essentially van der Waals interactions and electrostatic interactions. So this energy function is then used to integrate Newton's second law, and you can predict the position r in time.
So, does gravity play any role?
No, gravity is very, very small in these.
Okay, gravity is never.
Ignore it.
Yep.
Okay. Since we are doing numerical integration for a large number of particles for a very long time, we need a supercomputer, and we are, at least for now, blessed with Abacus, which is the largest machine in Denmark and probably in Northern Europe. And without it, you would not see many of the movies you are about to see. Okay, coming to applications now, I'll talk about three major applications.
I think you're doing very well.
So I'll slow down a little bit.
So, 16 minutes in the game.
Okay, so I have another. Okay, so I have enough time. So let's take a small break. Are there any other questions?
Yeah, can I ask about this Abacus? Is it just people from SDU using it, or whole Denmark? Or?
So anybody in the world can use it. They pay for it.
Mm-hmm.
But SDU, Aarhus University and Aalborg University have exclusive access to it because they help in building this computer.
Mm-hmm.
So researchers at these three universities have access to the computer.
Mm-hmm.
Right now it is not without, it is without cost.
Mm-hmm.
In the future this might change.
Mm-hmm.
More of like a sort of fundamental, like when I go to the lab, it's sort of expected that there's a possibility that you could make a mistake, and sometimes these might be happy accidents.
Happy accidents.
Like happy accidents where you actually find something interesting that if you hadn't made a mistake. But if something is so controlled, like in your experiments, do you ever have these sort of mistakes where?
Yes, we do.
Okay.
So we can also make these happy mistakes. I can promise you that none of the people who are doing simulations have not made these mistakes.
Okay.
They have all made the mistakes.
Yeah.
How many of them are happy mistakes? I don't know.
Okay.
I have not made a happy mistake. Most of them have been very unhappy. So during my PhD, I used to go on vacation every winter in India.
Mm-hmm.
And my advisor used to say, okay, you run these simulations before you go home, and I'll give you a 1 month vacation. So I used to run these. I used to start them and then go home. And one time this entire thing ran and I had no output because of one mistake that I made. But the simulations didn't crash, so we lost all the CPU power that we had. That's an unhappy mistake, but but we can have happy mistakes.
Okay.
It's the same.
It's possible.
So I'll talk about three topics. The last one is very technical, a bit more technical, and the first two are more applications based. So the first topic is flexoelectricity, which is the coupling between electrical potential and curvature in lipid membranes. And Dennis, who's seated here, is working on this. Before I go into that, I should introduce membranes to those who are not aware of what they are. Our cells, which are about a micron in size, that's 10 to the power minus 6 meters, are surrounded by a membrane which is about 5 nanometer thick. It's a 1,000 times thinner layer of oil which surrounds our cells and keeps them compartmentalized. This cell membrane is composed of many things, mainly proteins and lipids, but the basic structural matrix is a phospholipid bilayer, which of course is composed of phospholipids, which look like this. A typical phospholipid has two parts, one part which likes water and another part which does not like water. So if you mix phospholipids in water, they will not like to be mixed with water. They will self-assemble into certain structures, typically vesicles. So lipid bilayer is composed of these phospholipids. What you should also notice is that these head groups contain a dipole. So this is the predominant lipid in cell membranes, one of the predominant lipids. There is a positive charge here and a negative charge here. There are also other dipoles in the system, but this is the major dipole. So So we're going to talk about membrane curvature. Now this image of a cell is actually very, very wrong. Cells do not actually look like this. Most cells don't look like this. So if you look at a microscopic image of a cell, it will probably look a little bit more like this. So cellular membranes are highly curved. They are not as smooth as you see here. And this curvature has a very significant role to play in many cellular processes. I will not go through all of them. But for example, they are involved in membrane scission, in the shaping of organelles, like the endoplasmic reticulum. They're also involved in the activation of enzymes and sorting of proteins to certain parts of the cell. So for example, certain proteins prefer to sit in these corners of the cell because of curvature. And that has a functional role to play. But our membranes are also subject to a transmembrane electrical potential of about 100 millivolts. It seems like a small number, but the electric field, because of this 100 millivolt potential, is as large as you would have in high power cable lines. So the membrane is under a very high electric field all the time. According to the concept of flexoelectricity, which originated for liquid crystals a few decades ago, a potential applied across a membrane will alter the curvature of the membrane, or inversely, if a membrane is curved, there is an electrical potential across the membrane. And this is not a new concept. A lot of experiments and theory was done by Petrov in the 1990s. None of it is very, I won't say that. Okay, so here is a simplistic explanation. So this is my lipid membrane. It contains several dipoles, the dominant one being this dipole here. So in this leaflet, the dipoles are pointing in this direction, and in this leaflet they're pointing in the opposite direction. Now if I were to apply an electric field in this direction, across this membrane, these dipoles which are now aligned with the electric field will try to come together to lower their energy. On the other hand, these dipoles point away from the electric field, so they will try to go apart, which means that the density of dipoles is higher here and lower here, and given that the number of lipids is the same, this will bend the membrane. And that can be simply described by this equation. So this delta phi is the electrical potential applied across the membrane. Kappa is a bending rigidity, which is a material property of the lipid, and C0 is a spontaneous curvature. So there are two types of flexoelectricity, the one that I just talked about, so curvature induced by potential, or if a membrane is curved, then there is an electrical potential across it, which can be described by this equation. So C1 and C2 are the two principal radii of curvature. F is the flexoelectric coefficient, and epsilon is the dielectric coefficient of it. Okay. So the question is, can we measure this flexoelectricity in simulations and see how significant it is? For those who are doing experiments with vesicles or applying patch clamps across membranes, you should realize that this might be important. Okay, so to measure this, we need a simple model. All I need in my model is it to have dipoles in the two leaflets. I don't need to have information of every atom in the system. As long as I have these two dipoles, I'm good. The model that we used is a coarse-grained model. I'll come back to that in the next slide. But before that, I'll describe how we measure curvature. So this value C0 here. How do we measure it? We measure it by taking the first moment of the pressure profile. So this pi z is a pressure profile, lateral pressure profile across the bilayer. And that is the difference between the lateral and the normal components. This is what the lateral pressure profile looks like. So if you're traveling from here to inside the bilayer, at the bilayer interface there are these dipoles which repel each other. And therefore you have a positive pressure here. And look at the magnitude of this pressure, it is hundreds of atmosphere. It's a huge pressure. Right in the middle, there is a positive pressure because the chains are entropically repelling each other. They don't like to be confined in a bilayer. So there is an entropic repulsion in the middle. And again, it is symmetrical across the lipid bilayer. And this can be measured in simulations. There is still no way to measure it in experiments because there is no probe that can detect pressure, which is smaller than 5 nanometer and that can detect pressure across such a system. Okay, and this is our lipid model. So this entire lipid is modeled by the few particles which contain a head group dipole. So this is what is most important in this model. There is a head group dipole. This model is called Martini. It's developed by Sievert Jan Marink in Holland. And then we take this model, we construct a membrane, and we run simulations at different potentials. And this is the first result. This is the lateral pressure profile at different voltages across the lipid membrane. And you can see that the profile becomes asymmetric as you apply a larger and larger potential. This is because the dipole density differs in the two leaflets now. If you measure the total dipole moment along the bilayer normal, so along this direction, its magnitude is increasing for the upper leaflet and decreasing for the lower leaflet. The scale here is minus and here it is plus. So this is as expected, as it should have happened. And this is the main result. So on the y-axis is the product between bending rigidity and spontaneous curvature as measured from the first moment of the lateral pressure profile. On the x-axis is the voltage. Each point here is an average over 50 simulations, 50 independent simulations. And we have to do 50 simulations because pressure fluctuates a lot in these simulations. And we find that there's a linear dependence between this product and the electrical potential. So what does this mean? This means that now, based on this model, I can predict if there's a certain electrical potential across the membrane, then that membrane will be curved by a certain amount. So changes in potential difference curve membranes. What this also means, if the other effect is also true, is that if you're doing an experiment with a 100 nanometer vesicle, there's an electrical potential across this vesicle, which is not driven by ion gradients. And this potential will be different for a 100 nanometer vesicle and a 500 nanometer vesicle. And this can have very significant implications for highly curved membranes of cells and proteins sitting in cells which respond to this potential. And this includes all the ion channels in our neurological system. All we know about neurobiology, a lot we know about neurobiology is based on these experiments where you take these proteins, you clamp the membrane, apply a circuit and measure the currents as you change the voltage across the membrane. Right? But this voltage can have two effects. It can either change the open and closed states of these proteins, or it can alter the curvature of these membranes, which can affect the conformations of these proteins. And the second effect is often, has not been recognized so far. Okay, that was part one. Any questions so far?
When you're making the curve model, I don't know how the biology of membranes work actually, but do you have the same amount of molecules in the inside and in the outside, and is it important?
Yes, that's a very good question. So, in a typical vesicle, the number of lipids will be different in the two leaflets. For these models, we are not simulating a curved membrane. We have a flat membrane, which has the same number of lipids in the outer and the inner leaflet. And then we are able to measure how much this will curve using some physics. Yeah? But in a real system, the number of lipids will be different outside and inside. That's true.
But what about the other molecules that typically are in biological membranes, proteins and proteins with sugars?
Yes. So proteins with sugars. So the thing with lipids is it falls in a category of a material which is a liquid crystal, okay, which is essentially a material which has a dipole moment which can realign with an electric field. It has a director vector that realigns. Proteins will not do that because they have a complicated distribution of these dipoles.
But they will impact the lipid bilayer and its behavior, wouldn't it?
Certainly, because most of the 40% of the bilayer is protein, so that will affect the bilayer.
You mentioned not to make your life even more complicated, but most cells, they have a very extensive glycocalyx, so there's like a almost mechanical dampening of any sort of membrane dynamics, but just since you have to move this layer as well. And this is of course only primarily on one side.
On the outer side.
On the outside. So how is that?
So I don't know how much this glycocalyx damps the fluctuations in the membranes. Yeah. But this phenomenon does not depend on fluctuations in the membrane. So the fluctuations in the membrane will maybe affect the bending rigidity of the membrane, the material property. But the potential induced curvature will depend only on the potential and on the curvature. Also, intracellular organelles, intracellular membranes do not have this glycocalyx. So I think the glycocalyx will typically affect this number, kappa, the bending rigidity. Anything else? So the second example is a bit more biological, and I'll talk about amino acids and so on. So please try to focus. I have noticed that people tend to, especially the chemists and the physicists, tend to lose focus here. So we'll talk about the molecular basis of diseases in the sodium-potassium pump. The sodium-potassium pump is right now using more than half the energy that your brain is using, if you have any energy left in your brain. Our cells have an ionic gradient across them. So we have a lot of sodium outside and very little sodium inside, a lot of potassium inside and very little potassium outside. The cell also contains ion channels, potassium and sodium ion channels. So this concentration gradient would be equilibrated if not for the presence of active transporters which use energy to maintain these gradients. These gradients are important for signaling, for electrical excitation, for energy storage and so on. So we would be dead without these ionic gradients. So here the transport is passive along the diffusion, it's just diffusion. Here it is active, it is using energy to transport against a concentration gradient. This is what the sodium potassium pump does. It consumes energy, chemical energy from ATP, and it uses this energy to transport 2 potassiums inside the cell and 3 sodiums outside the cell. Now this poses several different problems for the pump. Imagine if you are the pump, you are facing the inside of the cell, and you need to bind 3 sodium ions, but all you see is potassium. You hardly see any sodium, and yet you must bind sodium. Okay? Let's say you somehow design yourself to do that. You bind sodium, and then you are facing the outside, and now you must release this sodium and bind potassium, but now there is only sodium outside. There is very little potassium outside. So this ion pump is selective to sodium on one side and potassium on the other side. Okay? So one of the focus areas in our labs is to figure out how this pump is ion selective. Different on one side, different on the other side. But I will not talk about that. What I will talk about is some molecular basis of diseases. So this is the structure of the pump. This is outside the cell, this is inside the cell. This is where the ions bind. These parts are catalytic regions. We will not talk about them, so you can forget about them now. This is the C terminus of the pump, and it folds back into the pump. And several mutations which cause diseases. So most mutations in this pump will kill you, because it is such a crucial pump. So most mutations will not accumulate over evolutionary timescales, but some do survive and they cause diseases. So these diseases are typically caused by mutations either here, where the ions bind, or here, which is the catalytic site, or in this region of the protein, which is the C terminus. So we're trying to figure out why mutations in the C terminus cause these diseases, and for this we have a collaboration with Aarhus University. So as I said, most of the energy in your brain is now being consumed by this protein, and we're trying to figure out why mutations in the C terminus cause these diseases. So what we do is we take this sodium potassium pump, we embed it in a membrane, surround it with water, and we run a simulation. So here is a simulation of a sodium pump, and you see some ions floating, and they will eventually bind the pump spontaneously because the electrostatic forces are very large. This is just an example. I won't talk about this movie anymore. And here we are zooming into this region of the pump. So we take the pump, we carry out a simulation of the wild type pump, and then we carry out a simulation of the mutant pump, and then we compare the two. So we are zooming in into this region. These are the ion binding sites. These purple spheres are water molecules, and this is a specific amino acid that I'll talk a little bit about. On the left is the wild type, on the right is a disease causing mutation. And what you eventually see is that in this case, water molecules are able to enter up to this amino acid only in the mutant protein and not in the wild type protein. This amino acid is aspartate 930. It's a negatively charged amino acid. And if you look at the hydration profiles of this amino acid, in the wild type, it is like this. In the other cases, water is much closer to this amino acid. And this is a simulation snapshot. This is an average over many simulation time frames. It turns out that a mutation in this amino acid in particular causes severe forms of Parkinson's. In electrophysiology, which is done in Aarhus, it was found that this mutation eliminates all current in pumps. I'm not showing you a comparison of the wild type, I'm sorry about that. If you measure the pKa of this amino acid, it is very high, so it should be around 3 or 4. This measures the extent to which it is ionized in water. This particular amino acid has a pK of about 7.5, so it is actually protonated. So based on this, a lot of other electrophysiology and a lot of other hand-waving arguments, we proposed that this amino acid is a proton shuttle. This is the typical mechanism that people thought of before our work. The sodium pump throws out 3 sodium ions on the outside, 2 potassium bind. The 2 potassiums are released on the inside via this pathway, then 3 sodiums bind, and the cycle keeps continuing. What we propose is that there is another pathway which is controlled by the C terminus. And in this, through this pathway, protons enter the binding site and lead to the release of sodium ions. And of course, there's a large leap of faith. I have not shown you all the data. I'm just trying to communicate what is possible to do. Yeah? So this work combined with electrophysiology led to the proposal that there's an extra pathway in the sodium-potassium pump. And if you mess with this pathway, it will cause you diseases. We have run some further simulations on some other diseases. So one of them is called KPOS. This affects very, very few people in Europe, but it leads to audit, hearing impairments, and so on and so forth. And the other is in hypertension individuals. Oops. So for this particular disease, you see exactly the same phenotype. So this is that amino acid, and again, water molecules enter in the mutant. And here also you can look at different parts of the protein, and you can focus on the molecular basis of why these diseases are behaving the way they are, or what is causing the diseases. How much time is it?
5, 6, 7 minutes.
5, 6, 7 minutes. Okay. So the last part is a bit more technical. It's about accelerating simulations. The problem that we have now is we can simulate after using 200 CPU years on the computer. We can simulate for a few hundred nanoseconds, and this is not good enough for many things. So we have to somehow accelerate simulations. The problem is that biological systems are hydrogen rich. Lipid has 52 heavy atoms, carbons, hydrogens, carbons, oxygens, and so on, and has 80 or 80 hydrogen atoms. The same is true for proteins. And the bonds containing hydrogen vibrate very, very fast. So my time step is limited to about 1 femtosecond. Yeah, that is 10 to the power minus 15 femtoseconds. That's not large enough. So how can we increase this time step? One way people do this is to apply constraints. So here, I will say that the distance between my carbon atom and my hydrogen atom is fixed to a certain distance. So this bond is not allowed to vibrate during the simulation, right? And that can increase my time step to about 2 femtoseconds. What else can we do without losing information about the hydrogen is use something called virtual sites. So in virtual sites, the hydrogen is typically treated as a massless dummy particle. The mass of this hydrogen is transferred to the nearest heavy atom. Remember that the hydrogen still has van der Waals interactions, electrostatic interactions. But because it is a massless particle now, it does not have bonded interactions. It has no bond, it has no angle, and has no dihedral distributions. So these interactions are completely lost for the hydrogen atoms. And then after the next time step, you can build the positions of these hydrogens using simple geometric rules. Right? So the position of this particle can be built from the positions of these particles. What this does is it increases the time step from 2 femtoseconds to 5 femtoseconds. It has been done for proteins, and now we are trying to do it for lipids, because lipids are also very hydrogen rich, and we are interested in different lipids all the time. So this is one example. This is the this is the tail of a lipid. These are the 2 hydrogen atoms, and in this case, they have been replaced by a simple dummy atom. And the positions of the hydrogen can be calculated from these simple equations. And then you can measure different properties of the lipid membrane. In this case, you can measure the area per lipid and the thickness, or structural properties of the membrane. And we show that with and without virtual sites, these properties are fairly identical. Similarly, you can measure order parameters. This measures how fluid a membrane is as you go from the, how disordered a membrane is as you go from the interface between lipid and water to the center of the bilayer. And the curves with and without virtual sites look fairly similar. And we are now trying to do it for all possible lipids. So this will, I'll skip this slide. This will essentially accelerate simulations of membranes by a factor of 2 or 2.5, which is a very big number. So if this works and if people start using it, we can save a lot of computing time. Or we can use the same computing time to simulate longer, depending on which side you're on.
does that on a sort of on a very large picture affect your sort of possibilities to actually get to the core of a biological phenomena when you're working on a time scale at best at nanoseconds, but you are well aware of that, of course, many processes are milliseconds,
Yes.
often seconds,
Yes.
but in some processes even longer.
Yes.
So there's a huge gap between time scale in biology and time scale of simulations, where you might simulate a very long time and still you don't see a single event of what would be.
So here you have to be smart. So you can do two things. One, you have to be very careful about, before you run the simulation, that you look at the time scale of the phenomenon you're looking at. So if your time scale of a phenomenon is occurring at much longer time scales, then you better not simulate for a much shorter time scale. Or you use techniques to accelerate sampling. Right? So in this case, for example, we are representing every atom by a single particle. But sometimes you're not interested in that. Sometimes 4 atoms can be combined into a single particle, and then you can bring out simulations for milliseconds. Or you can use techniques for accelerated sampling that I have not talked about. You can do something called replica exchange. So the idea is to explore the conformational landscape as much as possible. But there's only a limit of how much you can do. So I cannot fold a large protein spontaneously in simulations. You're perfectly right, it's a major limitation of 1D simulation. But there are other methods you can use that. Okay. I'll take a couple more minutes to show you some snapshots of additional examples. We did a small project with Samsung where we designed an artificial membrane made of these 2 surfactants. And this membrane is only 2.4 nanometer thick. This is the thinnest membrane that I am aware of. And it is very robust. So it is highly ordered, and it is much more robust than a lipid membrane. I don't know what Samsung is doing with it. We have used some quantum level calculations. Now I am not an expert on this, with collaborators in Japan, to design chemicals which can extract radioactive cesium from soil. This was done after the Fukushima disaster. So they developed radioactive chemicals, and now you can grow plants in that soil and put these chemicals with, and you can extract radioactive cesium from these soils. This we can skip, since I've already shown you a self-assembly simulation. So here is a self-assembly simulation of 2 different types of lipids becoming a membrane. This is what should happen. And finally, a short summary. I hope I have communicated to you that MD can be a powerful tool to investigate biological systems, as long as you are careful about the problem you are trying to investigate. It provides both visualization and thermodynamic quantities. In our group, we used MD to investigate different phenomena, but basically things to do with membranes and happening around membranes. And I've shown you 3 examples from that. And I'd like to thank you for your attention and thank everyone involved in the process. In particular, I'd like to thank Memphis, where some of the ideas have originated, and without that environment, I would not be around for so long. Thank you very much for your attention.
Thank you very much, Himanshu, for this very clear presentation. Any further comments or questions?
Maybe one which is a particularly sort of technical one. You showed us some options to sort of, by using a better methodology to accelerate. How does that scale comparison to GPU acceleration, where you have sort of very dedicated hardware to sort of implement some of your sort of mathematical steps? What is
So
adding
GPU
more
acceleration, no, both are important.
Yeah.
So GPU acceleration is hardware based. What I'm talking about is algorithm based.
That's
So
why
both these advances are important, and both work on GPUs. So the algorithms should work on GPUs, and if they do, you'll get a better scale.
But what is scaling faster for the time being?
GPUs.
Yeah.
Yeah, certainly. If you look up this January issue of Nature, they have a lot of talk about quantum computing. So I mean, in 5-10 years maybe, if Hanson tells us something.
So you told in the beginning a little bit about the limitations of experimental work. And we have seen some interesting results, but you also yourself mentioned some limitations of
Yes.
the theoretical work, or the modeling work, simulation work. You mentioned in your CV that you work a lot together with experimental scientists. So how do you work together?
It's a very good question. They tell us what to do and we do that. No, I'll give you one example of it. So for this problem, the sodium potassium pump, for example. So if you, I don't know if you believe me or you won't believe me, but
I would believe you.
okay. So after we ran these simulations, after we figured out that this pathway opens up and it reaches this amino acid, this mutation was discovered after that simulation, not before that. And once we discovered this particular phenomenon, our collaborators did electrophysiology experiments on this mutation, not before that. So we can suggest mutations to them which we think may be relevant from simulations, and then they can implement those. So this is one example of how we do it. In some other cases, somebody might come to us saying that, okay, this is a phenomenon I can't understand, and can you give us a molecular level picture of what might be happening? How does this drug bind the membrane, and why does this drug bind better than the other drug?
Okay.
And I should also mention some other limitations of the method. Our force fields are not polarizable, which means that the partial charges on every atom are fixed in time. This is not the case in reality. They will fluctuate a lot depending on their local electrical environment, and this is a major limitation of this method. However, for many cases, such as the ones I have talked about, this is not a problem, but it can be a problem from some other cases.
I just ask one quick question. The structure of the ATPase that you have, where do you have it from? Is it like already solved by somebody, or can
Yes.
you? Okay, and so you're limited to kind of the structures or analogs that are
For
already
proteins,
solved.
yes.
For proteins, yes. Okay.
What is your limitations in terms of molecular size? And I will come with a very concrete question. We in the Bionic Center, we have worked on fusion of liposomes, and of course this is something which in nano square meters is much larger than an individual protein, even a channel protein, which is usually very large. Is that accessible if you would have to look at patches 10 by 10 nanometers, which is of course huge in terms of how many lipids and atoms are involved in?
10 by 10 is not very big.
Yeah.
Even 20 by 20 is not very big. But it depends on the question you're trying to answer. So if you're trying to answer, okay, what should I design this DNA-based linker? How should I design it? What bond should I change? That I cannot do. If the liposomes are 50 nanometers each, or 30 nanometers each. It completely depends on what you're trying to ask. So if you're looking for the fusion process itself, you can use a coordinate model and you will see fusion. So fusion has been extensively studied in simulations of different resolutions. So you have to ask me a more specific question before I can answer. I think we talked about it many years ago.
Yeah, but that was before the fusion was actually happening.
And maybe also the simulations have been quicker and better now. So
Absolutely.
Yeah. And also Himanshu is much faster now than we are.
Okay, further comments or questions? Thank you very much, Himanshu, for giving
Thank
this
you.
talk and
It was a pleasure.
taking part in the discussion. Thank you.